I'm relatively new, assistant professor in the Department of Molecular Physiology and Biophysics at Baylor College of Medicine. Hassan's main interests are in algorithms, machine learning, and comparative genomics, typically relative to the molecular mechanisms underlying transcriptional regulation. However, Hassan or Dr. Samee has agreed to give a seminar to us on some of the basics of machine learning. And this is a precursor to the symposium that we will be having on November 10th, the Translational Imaging Symposium as it relates to machine learning and artificial intelligence. So I would highly encourage everybody to consider registering for that. We have a really good lineup of speakers on the agenda. But getting back to Dr. Samee, he received his PhD in computer science from the University of Illinois Urbana-Champaign, and he subsequently did his postdoctoral fellowship and research at UCSF at the Gladstone Institute. Dr. Samee is the recipient of numerous awards, including a Fulbright Science and Technology Fellowship, the Mavis Future Faculty Fellowship, as well as the Gladstone Institute Award for Scientific Leadership. I myself have found him to be a superior colleague in terms of collaboration and novel ideas, and he is starting to apply some of his skills in machine learning and artificial intelligence towards imaging on some of the projects ongoing within my laboratory. So we are absolutely delighted to have you here, Dr. Samee. Thank you so much for agreeing to speak to us, and I will then hand it off to you.
Thank you very much, Robia. That was really a kind introduction, and it's my great privilege to be able to speak to all of you. So ideas from imaging has been used for quite some time in genomics, and it's actually going to be great to talk about some of the new projects that we are starting here at Baylor College of Medicine on spatial transcriptomics, where we are actually borrowing even more ideas from imaging. So today's talk, as Robia said, that in today's talk, I'll try to describe how we are using machine learning to understand transcriptional regulation. And in that process, I'll spend more time on explaining our models and approaches and less time on discussing about some of the highlighting results that we have got. But I hope that that will be a useful bridge between me as a new member to this community. So this is my outline for the talk. The first part of the talk, I'll stay focused on some of the new things we have done in our lab at molecular level to understand transcriptional regulation. So this is the general picture that we have in our mind when we say that we are trying to model transcriptional regulation. We talk about a gene being transcribed. So in this cartoon, these ribbons, they are the mRNA copies. And the transcription level of this gene, the number of these mRNAs, they're controlled by regulatory DNA sequences, commonly known as enhancers that I'm showing here. And this function is carried on by transcription factors or regulatory proteins that I'm showing here as TF1 and TF2 transcription factors. So TFs, they bind to specific sites on enhancer, and when they bind, they regulate gene transcription. So the two fundamental questions that we're interested in are, first of all, how do transcription factors recognize their target binding sites? And secondly, when they bind the DNA, when they bind at enhancers, how do the DNA-bound transcription factors regulate gene expression? And regarding the first question, what I'll talk about is a role of DNA shape in specifying the target sites for transcription factors. So transcription factors, classically, they're understood as recognizing sequence patterns or sequence motifs in DNA. We'll come to those very soon. But what we have found using machine learning based models is transcription factors actually recognize DNA shape to an extent that goes beyond sequence motifs. So this is an example, and again, I'll come back to this example in detail, but this is what I mean when I say DNA shape-based mechanisms going beyond sequence-based mechanisms. So here I am showing the sequence motifs for two transcription factors, JUN-B and FOS. Both of them are known to recognize very similar sequence pattern, TGA, and then a fairly non-specific nucleotide at the middle position, followed by TCA. FOS is technically the same. So this is kind of a confounding observation that if they recognize the same DNA sequence and if they're expressed in the same cell, how can we see in genome-wide transcription factor DNA binding data, how can we see that they are binding at distinct locations? So what our model discovered was actually at the DNA shape level, their preference change, and the DNA shape motif, I'll call them DNA shape motifs, or these transcription factors are entirely different. That's the kind of pattern that I'll show in the first part of my talk. And regarding the second question, how do DNA-bound transcription factors regulate gene expression? I'll talk about a new model that we have developed in our lab recently, which we are calling multinomial convolutions, that can jointly model the DNA binding specificity of transcription factors and their effect on transcriptional regulation. So this is the classical convolution network that we have seen many times in news articles doing amazing things. And in fact, even in genomics, the convolutional neural networks have done amazing things. There are, of course, some outstanding issues. And what we will show is if we take each convolution is a matrix. And if we take each convolution and apply a certain simple, well-known transformation on that convolution, this actually gives us a much simpler linear regression based model, which is mechanistically explainable and which also avoids a bunch of other headaches in terms of interpreting the model. So these will be the first two stories that I'm going to talk about. And then the third part of my discussion will be more related to some of the imaging algorithms that we are now studying to study transcriptional regulation in spatially resolved single cells. So this is the type of data that I'll be talking about. We will get data at a level of single cell. We'll get gene expression data at a level of single cell. And with each cell, we'll also get its XY coordinate on the tissue. So the data matrix will look like this. For each cell, we'll have expression value for the genes in the cell, along with the spatial coordinate of that cell. So this is a rich data source. And looking at this data source, the first question that we are asking are, which molecular aspects of a tissue's function depend on spatial variation in transcriptional regulation? And just as a quick summary, we will show that with spatial variation in transcriptional regulation, we'll show interesting patterns of changes in composition of cell types, in co-localization of cell types, and in intercellular communication. And I would say that although I have phrased the question here with which, our ultimate goal is to understand why and how. But as we just started on this type of data set, we are understanding that what we first need to identify is which molecular aspects are quantifiable as being changed under variation of transcription and regulation. So most of these three things are basically our way to quantify changes in these molecular aspects. And eventually we are starting to move to compare these changes. Once we have quantified them, we want to compare these changes between healthy and diseased or healthy or injured tissue samples. So those will be the three main things through which I'll share some of the machine learning models that we have developed in our lab. So the first story is about transcription factors recognizing DNA shape beyond sequence motifs. And in general, again, this is, I guess, very much known to everyone that transcription factor DNA binding is understood as a sequence-specific mechanism. So in this cartoon, I show that in this DNA strand, this red transcription factor identifies this particular sequence ACAGTCA as its binding site, and it binds there. And in general, the preference of transcription factors to identify their specific target sites are represented as this type of models, which also called a motif or logo, which essentially denotes the same thing, a pattern of sequence. So these are called sequence motifs. And what it tells us is this particular transcription factor prefers an A very specifically as the first position, a C very specifically at the second position. It is okay with a G or a C at the fourth position, and so on. So these motifs, they essentially represent a probabilistic model that I'm showing here as a matrix, which tells us that if the transcription factor binds to a particular location in the DNA, what is the probability to observe an A at the first position at that site? What is the probability of observing a C at the second position? So these are all probability distributions essentially. So for like classically, this problem has been studied based on sequence motif, and the mechanism is understood as sequence specific mechanism. However, we have seen in our analysis of genome-wide transcription factor DNA binding data, and also in very well controlled in vitro transcription factor DNA binding data, that sequence preference alone cannot explain transcription factor DNA binding in many regions genome-wide or in many sequences in vitro data sets. So based on some classical hypotheses and some recent experimental studies, we hypothesized, so this is kind of the situation we often see, that this same transcription factor, it recognizes a completely different binding site at some other parts of the genome. So typically it recognizes maybe AC, AG, TCA. That's what we also found in the literature. But in binding assays, we find it recognizing some additional binding sites. So as I was just saying, that based on some recent experimental observations and some classical studies, we hypothesized that maybe this transcription factor, for this transcription factor to recognize its binding site, it's not only the sequence that it is trying to recognize. It could be also the structure of the DNA that it is recognizing. So if we plot the value of role, which is like the angular deviation between adjacent base pairs where this transcription factor binds, if we plot the value of role, it turns out that the value of role is very similar for these completely distinct sequences. So that's how we started thinking that can we build a model to identify the DNA binding preference of transcription factors in terms of DNA shape. So before I go into the model, I'll just spend one slide to tell a little bit more about DNA shape. So typically in the computer files or in the textbooks, we write DNA sequence as a linear sequence of A, C, G, and T, but it is a three-dimensional molecule. It has this backbone and it has the base pairs. And we know that there are angular displacements, there are linear displacements between bases and between neighboring base pairs. So these angular and linear displacements between bases and neighboring base pairs, those are the measurements that we call shape features. And those are the things that actually define the conformation of DNA, the DNA geometry. And anecdotally, we know that 4 shape features are known to influence transcription factor DNA binding pretty heavily. So in our initial study, we have actually focused on 4 DNA shape features, which are helical twist, minor groove width, propeller twist, and roll. So when I said that I'm going to plot the value of roll for each position across this DNA binding site, we are talking about the angular deviation between the 2 neighboring base pairs. And there are very well accepted computer programs right now based on simulation models that can give us pretty accurate measurements within a small DNA length scale. So, and since we are looking at a small DNA length scale anyway, these approaches fit very nicely to our need. So that's how we came up with the hypothesis, and that's our goal now, to identify these similar patterns of DNA shape within the sequences where a transcription factor binds. And we call them shape motifs, and we suggest that there is a shape-specific mechanism in transcription factor DNA binding. So this is an outline of our algorithm, and I'll try to go in smaller steps. The algorithm, its objective is to identify DNA shape patterns that are overrepresented between positive sequences, meaning where in the binding assay we saw the transcription factor binds, and negative sequences where we saw the transcription factor does not bind. So to identify the shape patterns, we first change or transform the sequences into their corresponding shape values. So once we change them into shape values, they look like this kind of noisy zigzag shape values. And our objective is to identify small DNA windows. By window, I mean small stretches of DNA within each of these sequences where the shape values are highly similar to each other. And to that end, we apply an algorithm called Gibbs sampling, which I'm going to explain in the next slide. So here now I have separated the DNA shape values for 3 sequences where we know the transcription factor has bound. But these are relatively long sequences, 100 bases or 150 bases long, and the binding site is hiding somewhere in these sequences. And our job is to identify those binding sites by looking at or by discovering locations where the DNA shape value is very similar. And the Gibbs sampling algorithm solved this problem through an iterative approach. So the sampling algorithm says that we'll start with a random set of windows or a random set of locations within each sequence. So this is just our first initial random guess. So the initial random set of windows, 1 per sequence, I'm showing those with those black boxes here. These are entirely random. And then iterative improvement is to try to find a better window for 1 sequence, assuming that the other windows are very good, are very similar to each other. So that's what I'm showing here. I'm now trying to update this window from this location maybe to some other location, but every time I'll try to minimize the difference of that window with these other given windows, because I'm assuming that they are good, they are fixed. So I'll start from.
Sorry, how do you fix the window size?
That's a good question. So we start with 10 base pairs, which is a typical length scale for transcription factor binding sites, but we also run separate, we also execute the same algorithm separate times with separate length scales. And finally, we have a model selection stage based on a validation data set where we find the optimal length scale.
Thank you, yes.
So, but this is a very simplistic explanation of what we are doing under the assumption of 1 fixed length. Let's say we are taking the length as 10. So all of these boxes have with 10 bases. But that was a good question. I mean, it would be clever if we could vary the length also within these sampling steps. So in terms of updating, as I said, that initially I'm assuming that these 2 windows, they're very good, they're very similar to each other. I just need to change this one. So I'll start from the left end of this sequence, and I'll gradually shift this window. Every time I'll see what have I got under this new location and how similar is it to the existing windows in my solution. So I'll try to find out more which one is the best. I'll do that. And maybe at some point I'll say that, okay, this is the best location for sequence 1. Now I'll say that I'll fix sequence 1 and sequence 3, and I'll try to update the window for sequence 2. And we do that until we converge, meaning that we do not think that any more update is necessary. So this is the essence of Gibbs sampling algorithm. And when we perform the updates, this is a probabilistic update. I'm not going into the detail, but this update procedure, it actually makes sure that another theoretical condition necessary for Gibbs sampling called detail balance is satisfied. So this algorithm, it is guaranteed to take us to a locally optimal solution, which may not be the best solution, but it is some kind of local solution when we converge. So that local solution will look like this. We will select this Gibbs sampling based algorithm will select 1 sequence or one part of the sequence from each of these inputs. And this is our initial solution. But we see that in this initial solution, some sequences have contributed a window where the shape value does not look very similar to the other shape values. So then we go to a second step, an outlier detection step, where we can prune out these type of examples. And at a very like intuitive sense, we just identify those examples which are significantly far from the mean and given the standard deviation of the current solution. So at the end of this procedure, we find this type of a pattern where at each position we are denoting what is the preferred value for the transcription factor in terms of angular measurements for this DNA shape feature. And this is the mean value, and here are some wiggle rooms. So this is what we call shape motif. And we did that for hundreds of transcription factors, and we found several interesting results from that study. Going back to this one example that I discussed at the beginning of my talk, was how do transcription factors within the same family recognize distinct shape motifs? And we found out very clear evidences that different transcription factors within the same protein family, and although they recognize in terms of DNA sequence, they recognize very similar sequences, their shape preferences can be very different. So we published this work last year in Cell Systems, and I'm happy to take more questions about this. But for the sake of time, I'm just jumping to the next part of our discussion, which is a second project where we are using machine learning models to understand if we are given data on the level of transcriptional regulation that enhancers can control, how can we model this data? In particular, this is how the data set looks like. So these are data sets coming from high throughput enhancer assays. In these assays, we give like thousands of sequences. We inject thousands of sequences to cell lines. And for each sequence, we ask what is the change in transcriptional regulation of a reporter gene. So every sequence is attached to the same reporter gene. And the readout, we call it the readout, the level of change in the expression of the reporter gene, we call it the readout. It's a normalized value in logarithmic scale compared to a control. So +2 means that this is 2 to the power 2 times change, or 2 to the power 2 times increase in expression in the reporter gene, because when it is put next to this sequence. And this is 2 to the power -3 times change, which is actually going down compared to control situations when the reporter gene is put next to this sequence. And by modeling, we want to answer a couple questions. First of all, what are the transcription factor binding sites in these sequences? So of course, it can be argued that we can pick up transcription factor binding sites based on publicly available motif libraries. But we do not want to bias ourselves to those motif libraries, and because we want to see if there are new motifs coming out from these analyses. And also, we do not always know whether the motifs, whether the transcription factors corresponding to those motifs are expressed in these cell lines. So we want to do this in a totally unbiased way. We want to identify the transcription factor binding sites. And we also want to say which transcription factors are inducive or activator of gene expression, or which one of them are repressive of gene transcription. So ideally, the solution should look like the following. We should be able to say that, or I should say our model should be able to say that in the first sequence, this is the potential transcription factor binding site, this green sequence, and this red part is another transcription factor binding site. In the second sequence, the green transcription factor binding site occurs again, and in the third sequence, the red transcription factor binding site occurs again. So we want to discover these patterns de novo without any prior assumption. And then we want to fit a model that takes some kind of scoring based on these patterns. It may be the count every time this pattern occurs in a sequence. It may be something more sophisticated, but some kind of score associated with these patterns. We want to take those scores, fit a model that can give us these kind of readout values when we input the sequences to the model. And also the model should be able to tell us which one of these binding site patterns look like binding sites for an activator or a repressor. And this is where we borrow heavily from the image analysis community, because image analysis community have like contributed a lot in the development of convolutional neural networks. So essentially we have to answer our questions, we have borrowed ideas from convolutional neural networks. So I'll spend the couple of slides here to discuss convolutional neural networks in a little in like very, very brief manner. So convolutional neural networks, they are well known for their power to discover small patterns in data. So this is one example of applying convolutional neural networks to recognize handwritten digits. So the idea is the model, a convolutional neural network, will be given any of these digits, handwritten digits, and the model should be able to say if this is a 0 or a 3 or a 9 and so on. And this is what a convolutional neural network typically looks like. I'm not going into the detail of the architecture, but as you can see, that typically these networks have to be very deep to discover those patterns. But essentially, when we say that a convolutional neural network is discovering a pattern, we mean that the network is actually learning matrices of values like this. So here we can see that this is a matrix of values, mostly 0, but there are some non-zero values at the middle of this matrix. And if we visualize this matrix as an image, it actually looks like this kind of a small, I would say, line stroke. And this is exactly what the convolutional neural network tries to figure out. It starts with this randomly initialized convolution matrices, and it keeps optimizing the values. So initially these are all random values, but the model tries to optimize these values to eventually find out these types of matrices where each matrix actually captures a small stroke in the handwritten digits. So in that case, we can see that these strokes, that they start modeling small parts of the input handwritten digit, and how to combine the existence of those small strokes into a definitive answer, whether this is a 0 or a 1 or a 7, that part is handled by this intermediate set of layers. So this is what we are kind of focusing on at this point of our work, that these convolutional neural networks, they're very good at discovering or learning these type of matrices that essentially represent underlying small patterns in the data. And these are like well-known examples from the handwritten digit recognition problem, that some of these convolution matrices, they recognize this type of a slanted stroke. Some of these convolutions, they can recognize this type of a relatively straight stroke, and so on. But again, as I said, that all these convolutions together, they recognize different parts of the input. And then the rest of the layers, they decide how can the presence of those different strokes can lead us to a definitive answer in terms of telling what should be the value, what is the digit? Is it a 0 or a 5? So this is where the genomics community felt excited about convolutional neural networks, because like an image, we can also encode a DNA sequence. So if a DNA sequence is ACTGC and so on, we can encode this into a matrix of size 4 by the number of bases in that sequence. And for every nucleotide, if it is an A, we put a 1 at the first row. If it is a C, we put a 1 at the corresponding column or at the corresponding base position in the second row, and so on. So every nucleotide sequence can be like uniquely represented as one of these matrices, which looks pretty much like an image to a computer program. And the hope is, if we can now input these representations of DNA sequence to convolutional neural networks, Our hope is, can eventually the convolutions start looking like this, so that we can say that I will look at the convolution and readily get the sequence preference for some transcription factor which prefers to bind at these motif occurrences. However, the convolutional neural networks by design, they do not mean to learn probability distributions over the 4 DNA nucleotides. In other words, the matrices, the convolution matrices in a convolutional neural network, they're entirely free to go up and below 0. And technically, they're free to learn any value. So what we have seen in the genomics community, that we need post hoc analysis. After we fit a model, we need to look at the model and we need to analyze the model to understand what has it learned. And those post hoc analyses, they often come back with very similar motifs that we have seen here, but it's not guaranteed that whatever motif is coming from the post hoc analysis is optimal for the same model. And also the models are very deep, at least 2 to 3 layers deep. And these post hoc analyses, they essentially interpret the first layer of the model, which do not lead us to any interpretable biophysical mechanism. And all these are basically, I would say, require more discussion. But for now, I just wanted to say that the state of the art of applying convolutional neural networks in genomics is finding this aspect of convolutions a problem in the first place, that we need to interpret the convolutions after we have fit the model. So what we wondered is, can we make the convolutions fit probability distributions over the 4 DNA nucleotides? And as I alluded to at the first part of my talk, that we figured out that it is fairly straightforward to do that for a given convolution. So if we are looking at a given convolution matrix X, if we apply a Boltzmann transformation, which is exponentiating each entry of X dividing it by a normalization value, which is the summation of exponentiated value of all numbers in the same row of that matrix. If we simply do that, it turns into a multinomial distribution, exactly what we wanted in the first place as the DNA binding specificity of transcription factors. And more interestingly, these multinomial distributions, they're very well studied. They have very well understood biophysical interpretations. So once we learn them, the rest of our model simply collapses to a single layer, which implements a linear regression model. So these multinomial distributions, we can then use them to compute likelihood terms. And then we combine the likelihood terms into a very interpretable, biophysically interpretable linear regression model to fit the gene expression. And these are some of the good things about the model. Again, I'm not going to dig into the detail of comparing the model against the other published models, but we really gained a lot of savings in terms of the number of parameters. And we saw that the transcription factor binding motifs that we learned, they're all relevant. We did not need to utilize any quote unquote non-mechanistic input into the model. And here is just one of the interesting applications that we found of this model. We took all the common variants in the human genome, and we wanted to ask which one of those common variants is potentially pathogenic or could do something deleterious. So we just asked our model that if this variant occurs in the input sequence, what is the predicted change in expression? And then we took all those sequences, we ranked them, and we found that the most deleterious sequences or the most deleterious variants predicted by our model, they actually are highly enriched with very low minor allele frequency values in the human population, which just tells us that using the model, we are finding the same signal that a negative selection would do, that potentially deleterious variants, they won't stay at high frequency in the population. And there are a couple other interesting things that we figured out using the model. And again, I would say that the most interesting part for us was this ability to tell the convolutions to fit specific distributions. And now we are taking the same idea to revise our GIP sampling based approach to discover shape motifs. Now we are just saying that to discover shape motifs, maybe I don't have to run the GIP sampling algorithm. If I can tell the convolution to learn a simple Gaussian distribution at each position of the binding site, maybe that is good enough. So that's what we are trying to do. But I look forward to getting more suggestions and applying this idea of specifying in the convolution the type of distribution that it should learn. That we see that adds a lot to the interpretability of the models. So that was the second part of my talk. And now we are jumping into the third part, where I wanted to show application again of neural networks in studying how spatial variation in transcription and regulation.
Cloud storage is full.
I'm sorry, is there a question? Okay. So in this part of the talk, I'm going to show a neural network model that we developed recently to study how spatial variation in transcriptional regulation impacts molecular and functional aspects of a tissue. So in particular, we are going to talk about composition of cell types, co-localization of cell types, and intercellular communication. And again, I mean, with like full disclaimer, I may say sometimes it's how and why, but by all means, this is our first attempt to quantify the changes, or to quantify how transcriptional, okay, I'm saying again how, I'm sorry, but these are the different aspects, molecular and functional aspects, where we are trying to quantify their changes with spatial variation of transcriptional regulation. So to begin with, this is the two data types that we need to answer to go into this pursuit. The first data type is single cell transcriptomics, or single cell RNA-seq, where we have cells from a tissue, and from each cell we have the gene expression values in that cell. So this is single cell RNA-seq. And then we also need spatial transcriptomics, where we get the gene expression values within cells along with the XY, the spatial coordinate of those cells. However, as I have told as I have mentioned in the title of this slide, these two technologies, they're complementary. First of all, for technical reasons, spatial transcriptomics is not good yet at profiling as many genes as single cell RNA-seq. So single cell RNA-seq, it loses the spatial context. It does not come back with XY coordinate information, but it captures almost twice as many genes as spatial transcriptomics can. So that's why I said that N, the number of genes profiled by single cell RNA-seq, is much larger than K, the number of genes in spatial transcriptomics. And then also we are looking at two different samples. If this is one tissue sample, we are getting one set of cells. This is another tissue sample. To show a little bit more about the detail of the spatial transcriptomics data, this is how it looks like. And this is one type of spatial transcriptomics data. Currently, spatial transcriptomics covers several different ways to profile gene expression at a single cell resolution. So this is one type of those data sets, seqFISH plus. In this assay, a tissue slice is taken. So this is from mouse olfactory bulb. A slice of the mouse olfactory bulb is taken. And then this is how it would look like. This is just a cartoon of the olfactory bulb slice. So in seqFISH plus protocol, we do not get data from the entire part of this olfactory bulb slice. We get data from some parts of this tissue slice, and each of those parts is called a field of view, or FOV, and this is the locations of the 7 FOVs that we got from these recently published seqFISH plus data. So in general, we got data about 10,000 genes, and in single cell RNA-seq of olfactory bulb, we get data about 20,000 genes. And all these 7 FOVs, they cover around 2,000 cells in the tissue sample. This is a slide I just put, but I should say that I'm really not an expert in the experimental side of seqFISH Plus. But very briefly, I just wanted to fill in the gap of how this technology assays gene expression. So with each mRNA, this technology attaches fluorescence probes. And so the probes, they actually constitute barcodes. And after 3 or 4 rounds of imaging of those probes, each mRNA produces this type of a barcode. And the barcodes, the combination of these colors define the barcodes, and they're unique for each gene. So then, counting the number of times each barcode is observed, we can get the count of mRNAs for that particular gene. So this is the basic idea I personally carry with me about seqFISH Plus, but if anyone wants to fill in, please feel free to do so. But as I said, the challenge in this data modality is we are profiling much fewer number of genes. So on the right here, I'm showing the data that we get from seqFISH Plus. This data tells us the location of each cell and the value of gene expression, I'm sorry, and the value of gene expression in about 10,000 genes in each cell. But the data does not tell us the type of those cells. And it's not easy to define the type of those cells just given those 10,000 cells. On the other hand, single cell RNA-seq pulls out a lot more genes, and we can very clearly cluster them into different cell types. So this is kind of the point where we have to somehow integrate the 2 data sets.
I'm sorry, can I just ask, why do you get so many more genes with single cell RNA-seq as opposed to spatial transcriptomics?
Again, so I should apologize that I am still learning the technical parts where the technology is limited. But as much as I know, since we are gathering data from a very small region of the tissue, there are some capture efficiency things. But I'm really ignorant beyond this point. So maybe I should get back to you about the details. No, I mean, yeah. So this is I'm trying to overcome, and it's kind of challenging because as a proof of concept, we started working on this data set, but there are several other data sets applying somewhat different technologies, but all of them face the same limitation. That's one thing for sure. So so with single cell RNA-seq, we learn the cell types very well. But again, with spatial transcriptomics, we know where it came from, and we know the XY coordinate within this FOV. We just don't know the cell type. And ideally, we want to somehow learn the colors of each cell and transfer that to spatial transcriptomics. So that's why we did. It's a very straightforward neural network model. The idea is we'll first learn the cell type of each cell based on the genes that are shared between single cell RNA-seq and spatial transcriptomics. And once we have learned those cell types, we can readily apply the model to predict the type of every cell coming from spatial transcriptomics. So this is our preliminary model. We have put the model and some of the interesting findings at the tissue level by applying the model, but we're actively working to improve the model. But in the rest of the talk, I'm going to present some of the interesting findings in terms of those molecular and functional phenotypes that we see changing with changes in transcriptional regulation. And by the way, this is the name of the model we made up, Spatial Transcriptomics Cell Type Assignment Using Neural Networks, STAN. The paper is available in bioRxiv. So as usual, we benchmark the model with a couple other like state-of-the-art models named SURAT and SCPRED, and the model did really well as compared to those. And as of now, I will say that we haven't done very clever things yet. We just changed the loss function to take care of imbalance in the composition of cell types. Like one cell type may be like very highly abundant in the sample, another cell type may be relatively rare. So we just applied a weighted loss function to optimize the model so that the model can give equal importance to all the cell types. And just by doing that, we saw that we are doing very well on rare cell types. But again, it's still a work in development. And very briefly now, I'm going to talk about these three aspects of the mouse olfactory bulb that we saw changing in interesting way. So first of all, composition of cell types. So in general, we saw that when we apply the trained STAN model on the cells in spatial transcriptomics data, STAN assigns types to those cells, and the abundance of those cells recapitulate very well with the abundance of those cells in single cell RNA-seq. I mean, there are minor variations in the rank, like neuron GC. We find that they are the most abundant in the spatial transcriptomic sample. They're the second most abundant in single cell RNA-seq. But in general, we see that the model captured the expected heterogeneity in terms of the abundance of different cell types really well. And then we went into studying the abundance of cell types within individual field of views. So again, these are the 7 fields of views. This is 2, this is 3, this is 4. And these 3 are all located within the same morphological layer of mouse olfactory bulb. And when we apply STAN tells us the types of cells within these 3 FOVs, and we see that they are statistically there is no statistically significant difference in the composition of these FOVs within the same morphological layer. However, STAN says that I would say the statistical tests says that when we go and compare one FOV and another FOV from 2 different morphological layers, the cell type compositions change widely, very significantly. That's true for example, 4 and 5, even though 5 have some part of it within the same morphological layer, we still see statistically significant change of cell type composition. So that is the first thing, that the cell type composition stays very similar within a morphological layer, but across morphological layers, they change. So the second question we asked was, okay, if the cell type composition is similar, do we see similar spatial co-localization of cell types? And again, this is a part of our work that we are still studying and trying to learn a lot from image analysis papers where co-localization analysis is fairly common. In our first go, what we did was we asked STAN to identify the type of each cell. So for FOV3, for example, we asked STAN to identify the type of each cell. And STAN said that these are the locations of neuron transition cells. These are the locations of olfactory and sheathing cells. We wanted to quantify their co-localization. So we said that, okay, let's try to find out the probability distribution for these cells to occur at each particular XY coordinate in these FOV. So we did something called a kernel density estimation. And once we did the kernel density estimations, it became straightforward for us to quantify the correlation between the probability values. And we did that. And even before going into the correlation values, what we found that although in FOV2 and 3, the composition are very similar, the co-localization pattern changes widely. In FOV3, they're highly co-localized, as I have also shown in this like more of a binarized version of their spatial occurrence. In FOV2, they are nearly exclusive in their spatial occurrence. And this is not only limited to these 2 cell types. This is common for almost every pair of cell types in these 3 FOVs, where the 3 FOVs are located within the same morphological layer. And we saw that the cell type composition is remarkably similar. So for example, we saw that for astrocytes and neuron GCs, in FOV2, they're co-localized to some extent, not to a high extent, but in FOV3, they're highly separate in their spatial organization. In FOV4, it's again somewhat separate. However, the things, as you can see, that they're changing. I'm not picking up specific examples for the sake of time. So this analysis was done by Zen, who was rotating in our lab at that point. So this is the second interesting observation that we saw, that the co-localization pattern of cell types change within the same pair of cell types and within the same morphological layer where their compositions are highly similar. And finally, we wanted to study a little bit into the mechanisms of intercellular communication. So we studied intercellular communication using expression of receptors and ligands. So we asked if cell type A and cell type B, if they are expressing a known pair of receptors and ligands, do they, I should say it this way, we used actually some published model which looks at the expression of known receptors and ligands in each pair of cell types. For example, receptor R in cell type A and receptor ligand L in cell type B. And those models do some enrichment test to answer whether this receptor expression in A and this ligand expression in cell type B is enriched. In that case, they say that A and B, they're probably communicating with each other using these receptors and ligands. However, those models were not built for spatial transcriptomics data. So we wanted to make sure that we are just not picking up those examples where A and B are really far and receptor R is actually being used by A to communicate with a specially co-localized cell type C, and ligand L is being used by cell type B to communicate with the specially co-localized cell type D. These would be potential false positives. So we checked for these potential false positives, we eliminated them. And then we came up with a list of receptors and ligands for each pair of cell type within each FOV. And this was actually very good that these FOVs, they were 20 nanometers by 20 nanometers by size. And this range actually already is kind of within the range of intercellular communication that was shown like more classically by Francis and Paulson in their studies on bacterial systems. So this was kind of the setup for us to study the receptor ligand based mechanisms for two cell types to talk to each other. And again, we found very interesting heterogeneity in this receptor ligand based communication. First, we saw that the same cell type pair uses very distinct receptors and ligands across the tissue in different FOVs. And secondly, I'm actually just keeping the plots for the sake of time. And secondly, we also saw the same receptor or ligand, it interacts with very distinct sets of genes in different parts of the tissue. So both of those were interesting to us, and we are trying to kind of use other data sets of mouse olfactory bulb to see if we find similar observations in those data sets. But I would say that those were the three interesting observations from this study on spatial transcriptomics. And now we are actually moving to a slightly different spatial transcriptomics technology, a commercially available platform called Visium. And our ultimate goal, as we have said in the beginning, is to be able to compare how composition, co-localization, and crosstalk or intercellular communication mechanisms change between a healthy and a disease condition, or across samples from developmental time points and so on. So at this point, I would really like to thank you for the privilege of your time. I also acknowledge my lab, my collaborators. The Spatial Transcriptomics project is a very exciting thing that we are doing in collaboration with the labs of Dr. James Martin and Dr. Diwakar Turaga. And please feel free to email me with your questions and suggestions, and I would be happy to take any question.
Great. Thank you, Hassan. That was a fantastic talk. I'd like to open it up to the audience if they have any questions.
Thank you.
Because I have a couple of questions, actually.
Sure.
And, you know, I guess one thing I'm wondering about the spatial transcriptomics,
Yes.
is that really limited? Oop, can you go to your olfactory bulb where you have the 7 FOVs? Yep.
This one.
So is this where the technology is currently at right now, do you believe, where you can only have a very small number of regions that you assess? Or would it be possible, you think, to make a matrix that spans the entirety of a tissue or organ or tumor?
As of now, I have seen all the studies going at like small patches within the tissue.
Mhm.
So I would say yes, this is the state of the art. Even the 10x genomics Visium that I mentioned, I forgot the exact length scale. That is probably bigger than these individual FOVs, but each Visium slide can actually take 6 or 8 small patches from a tissue. I can look up the numbers actually, and let me see if anyone in the audience I know can give a better answer. Yeah, sorry. I think I can say for sure that this is a limitation of the technology, that we cannot do single cell level spatial transcriptomics on an entire tissue. These are all the data sets that I have seen so far are coming from small parts of the tissue.
Okay. How big are the FOVs that you're outlining right here? Are they to scale in that image, or?
In this image, they are not to the scale. In this image, they are. And this is actually a mistake. This is 20 nanometers. So these are about 200 nanometers by 200 nanometers.
Well, Hassan, as you know, I'm also very interested in the olfactory system, and actually I would love to talk to you about a couple of ideas I have.
Definitely. I think this whole project was kind of a proof of the concept study, but we got like so much excitement from these two data modalities that any potential input or collaboration, I would love to go into that.
Okay. Could you go back to your olfactory bulbs with the 7 field of views?
Yeah.
Because I think, as for those of us who do MRI imaging, for example, we see these olfactory bulbs all the time and they look like that. But I guess I'm just wondering, in terms of applying some of your modeling to like imaging data, for example, how arduous of a process would it be if we could just, for example, give you imaging data per field of view, or voxel, as we should say, for the entirety of a structure?
And then what is the data within that voxel, like mRNA or protein?
Imaging output.
So those I think I'm trying to understand what phenotype are we recording there?
Oh, so I'm just saying, let's say we had an olfactory bulb, and in this olfactory bulb, we could do many different types of imaging, for example.
I see.
And from a specific field of view, you could get multiple outputs.
I see.
And then from that, so I'm wondering how arduous or complicated of a process it would be to apply some of your really cool modeling methods to really start to get a lot more out of the imaging.
So again, I'm just saying this based on my confidence on the developments in terms of machine learning on image data. I would say that technically any question that the data supports, we can tackle that question. In terms of the difficulty, I think we have the capability in our lab, although the lab is small, but I think we have the capability to try different types of models to integrate. If you need to integrate different types of information, we have the capability for that. But I think that the deep neural network community has made really tremendous progress in image analysis. So these are doable.
Okay, great. Are there other questions from the audience? Okay. Well, thank you so much, Hassan. This was really great. And I mean, it's kind of hard to applaud. I can do like my virtual applause right here.
I appreciate everyone's time. And again, I would request for any kind of input. Again, my email is samee@bcm.edu. Please let me know if you have any question or comment.
Okay, great. Thank you so much. And thank you to everybody for attending the seminar.
Thank you very much, Robia.
Okay, thank you, Hassan.
Thank you. Bye.